one concentration to the next, right? If the curve is really steep, right, his slope is going to be large. If it is very slow, his slope is going to be very small. Okay? Now, I know Addie, you wanna be in pharmacy, right? So we're opening up a compounding pharmacy to get peptides. Yeah, again, your next students in this lab are going to use a peptide that will dissolve fat. So we're going to put that in transdermal cream so that people can just rub it against, an area they want to dissolve the fat. It goes in and get the fat disappear, right? No. So, yeah, because all the Ozempic and all this stuff, they're very popular. Well, this one, it goes from outside in, right? Now, these are the numbers that are very, very important, right? That we need to get, okay? So, you do this, you do that, and then, so this is for the rescue. And you do exactly the same thing with the toxicity. Okay? You're gonna have full control, which is a bar graph. And for the toxicity, go ahead and calculate the percent cell death, right? You count it. Here's what we have, right? Here's what we had. We had clear cells and we have blue cells, right? The total should be around 20, but use the total. Yeah, so the numbers kind of look like this. There's clear, and there's blue, and they will give you the following. Let's say 19 and 1. Okay? So what you do is 19 divided by 20 is equal to the percent cell that survive, okay, right? And so it's going to give you 95%, which means 5% for dead cells. Mm-hmm. And that's your percent cell there. And put that in the table, right? Generate a table, okay? And then you tell it, generate a, once you do that table, you do the same thing, right? You paste it into Copilot and say, generate a table containing average cell death, right? We don't need full control for this one, but we need the standard error of means, and then p-value, okay? And it's going to generate a table of all that calculation. And then you take that table just like you do for the rescue, and you paste that in Copilot and said, generate a bar graph of percent cell death versus drug concentration. And it's going to generate that just like it does for the rescue, okay? Mr. No, you have a question?
What is fold control and why does the toxicity report not meet them?
Pardon me?
What is fold control?
Okay, does someone want to answer that? What is full control? It really is not a science term. It's just a term that means
how well the protein folds once that it has been gone through RNA, ribosome. How well the protein folds after it's been created, I guess.
Okay, yeah. Anybody? Full control. What does it mean to you? So, control is no drug, right? Mm-hmm. It's going to generate a certain amount of rescue. Mm-hmm. With the drug, it's going to generate a lot of rescue of the protein. So if you take the rescue in the presence of the drug divided by in the absence of the drug, that gives you a full control. How many times, right, you get multiplied over the zero concentration? Okay? It could be 1.5, 2, 3 times. Yeah, the drug concentration is stimulating rescue 1.5, 2-fold, 3.5-fold over the control
So
concentration.
How many times better than,
Pardon me?
So how many times better
No.
that drug?
How many times, how many multiple of control is resulted from the drug treatment.
Okay, sure.
Yeah.
Pardon? As in if you're,
I guess you could say it's better, but it's how many fold, how many times? Mr. No,
yeah.
how many multiple of control you generating with the, in the presence of the drug?
Okay, sure.
Okay? All right.
If it would be-
Okay, so, yeah, George.
So in the rescue data, so it includes the control value, and presumably the control value should be 1, or if it's how many times better than the control it is, shouldn't it?
Okay, everybody pay attention. So let's say that we do the concentration right here, right? So let's say 0, 50, 100,
Mhm.
right? The amount of rescue at 0 is, let's say, for lack of a better word, 0. Let's say 1. At 50, it's 2. At 100, it's 4. Make sense?
Mhm.
So these are the values in the presence or absence of the drug treatment,
Okay.
right? Now, full control is you divide everything by the control value, right?
Mhm.
So George, according to you, full control for at 0 is 1,
Okay.
2x, and 4x, right? That's what it means.
Mhm.
Or we can, hold on, we can change it to 0.5,
Okay.
right?
Mhm.
Full control at 0 is still 1. Full control at 50 is equal to, what's that equal to?
4.
4. And full control at 100,
8.
8. Mr. No,
Yes,
do the numbers make sense?
yes.
Yeah, how many multiple of the value at control you get with the drug treatment?
So how come we don't need this for the toxicity?
Pardon me?
Why don't we need this for the toxicity analysis?
No, we just need percent cell death and not the full.
Okay.
Now, let me say this, if you do full control, you're gonna get exactly the same thing. You see what I'm saying?
Yes.
Yeah, so, listen, we're gonna use this data, right? For the clinical trials and that we'll move forward on. For the toxicity, we don't use the full control.
Mhm.
That's why we're excluding, right? But we do need it for the rescue. So we don't need to calculate if we're not gonna use this. But if you want, you can calculate it and look at it. Okay? So for the toxicity, it's gonna generate a curve. And this is gonna give you an LD 50 value. Okay? What concentration of the drug will kill 50% of the cell? That's what it's gonna generate for the toxicity curve. Okay? So you have generated 2 tables and you have 1 graph, 2 graphs for the rescue, 1 graph, 2 graphs for the toxicity, okay? Now, here's why this is important, right? Because there's something called the therapeutic index. Which is equal to LD50 over EC50. Okay? Now, I told you all that we're generating a compounding pharmacy. So, we're gonna look at the drugs that you are working on, right? All of you are working on. The therapeutic index could go from 3 to 3,000. Okay? Now, the small window means that the toxicity and the rescue are very close together. Is that good or bad?
Bad.
It's bad. Yeah, that drug is not good, okay? Let's say that G418 therapeutic window might be 3.8, right? That's very low. And let's say ataluren therapeutic window is 3,000. That means that you have a large window to operate to treat patients within that large window before you reach toxic level, okay?
Right.
So that's what we're looking for. Yeah, this is what we're really wanting to see. Jordan.
Never mind.
Go
I
ahead.
figured it out.
No, go ahead.
I mean, I was just took me a minute to figure out why a higher number meant less cell death, because a higher number means greater concentration needed to get the lethal dose. That's took me a minute to figure that out.
Yeah, listen, if you treat the cells with, let's say 50, and you get 5% cell death, right? 50 microgram per ml, and you treat the cells with 1,600 micrograms per ml, and you're getting 75% cell death. Does that make sense? At low concentration, you don't have a whole lot of kill. At high concentration, that drug is so toxic for it. And in fact, at the 3,200 microgram per ml, you kill nearly 100% of all the cells. Okay? Yeah, so low concentration. So when we do clinical trials, right, you start with concentrations that will not affect individuals. So here's what we do in clinical trial, phase 1. So let's say that I want this ataluren. We want to test that for Bainbridge-Hoover syndrome, right? We're going to recruit healthy individuals, and then we're going to start with dosing them, right? For you, I may start with 10 micrograms per kilogram of body weight, right? We're going to observe you in the hospital, make sure you don't show any toxic effect. If it's safe, then we move on to next individual, and we do 20 micrograms. If it's safe, we go up to 40 micrograms. If it's safe, we go up to 80 micrograms. We go up until they show toxic effect, right? So now that's our window, right here. Yeah, where we don't see any toxic effects. Yeah, but if that window is very wide, that's good. But it could be for G418 that once we step up from the lowest concentration, you begin to see side effects. That's not a good drug. Okay? All right. Any questions? Yes.
So the presentation we're giving in class is like a poster we make based on our research in this class.
Pardon me?
The presentation we're presenting next week, is it like a poster presentation?
Oh yeah.
Okay.
Yeah, it's presentation on the data you've
Okay.
gathered, right? Yeah. All the data that you've gathered is going to be part of that presentation. And I gave you, hold on, I gave you an example, right? We're gonna go all the way back to the beginning of the lab. You're gonna get all that data, right? So, I'll answer the questions, but let me go back here. If you haven't seen it, please take a look, because it really helps all the directions, right? So, figure 1, figure 2, figure 3, figure 4. So this one is the vector and the cloning. Take it straight from the PowerPoint that I gave you. I'll upload it for you, okay? Yeah, cut that picture, paste it in there. That's figure 1. All right? What do you think figure 2 is? We take that plasmid, transform it in bacteria. What's figure 2?
Transformation.
Transformation. That's the transformation plates. Okay? And figure 3, what do you think figure 3 is?
Colorimetry.
Pardon me? Colony screening,
How can you fit this all into one poster? How can you fit that all into one poster?
all right? Figure number 4,
Restriction digest.
restriction digest. These are all the figures you have generated already. They are in your lab notebook number 1. You just cut and paste them in there. And make sure you have figure legend for each one of those, right? Let's see, and then, I think that's all from the first part. And then second part, you're gonna have table. And that's the rescue. And then you're gonna have that graph, right? Full control.
Is that the EC50 graph?
Bar graph. And then this is the full control. Dose response. Okay? That's just for the rescue. And then you come down here, you have table number 2. And then you have full control for average. Percent cell death. That is bar. And then percent cell death. And this is the dose response curve. Okay? See all the figures that you have? Yeah. All of these are from the lab notebook number 1, and all that are from lab notebook number 2. Okay?
I have a question. What's the difference between figure 1 and figure 2?
Between figure 1,
In figure 2?
figure 1 is the vector, the picture of the vector.
So like as in like an actual as in like a bio-rendered picture of like a vector.
Yeah, it's,
Oh, okay.
it's actually in the PowerPoint, yeah.
Okay, cool.
Where I show you how we clone that truncated gene.
For sure. Okay.
Just cut and paste that in.
Which
Whereas
figure is that?
figure number 2 is the transformation.
And then once again, this
Okay,
is
perfect.
so this is a poster or a PowerPoint presentation.
It's
that
poster.
We're sort of like.
Yeah,
Are we printing it out then?
pardon me?
For the poster, are we like responsible for printing it out? Because I don't really know.
If-
For the poster, are we gonna print it out? Because.
No. So here's, you've never presented poster before, right? I mean, in class. So what you're gonna do is, listen, I have uploaded the template for you on the Canvas, right? All you have to do is fill it in, right? You're gonna fill in that poster. It's sort of like you did for Summer Science Institute, right? Instead of printing it out, right? You're gonna bring it here, we're gonna project it right here, and you and your partner are gonna stand here and point to it, to the picture, instead of the hard copy. Okay? All right, we have Mr. Lamb.
The hardest part is making
So
the
in our
graphs.
second lab notebook, are we
can do that
going to
with
remove the preliminary trial parts? Cause that's what I saw in the sample.
And
Where
then
you put
No, no,
preliminary
I'll transfer it.
No,
to
no,
mine,
no,
'cause I
read the
haven't done
instruction.
Of this mine.
Okay.
And I'll switch some stuff.
That-
out.
They are there.
Listen. Here's what I said, right? The preliminary study, first part, is the common data for the rescue. The second part is the common data for toxicity. That's 6 and 7, right? And then when you have 8, you're gonna have 8A, 8B, or 8C, right? 9A, 9B, 9C, yeah. Does that make sense?
Yeah, but then.
So preliminary data has to be included. Table. Yeah, that's number 6. Rescue. Table, common data. That's table number 7. Toxicity. Okay? read that instruction, it's really very straightforward, clear. Yeah,
In the sample poster, it also includes sections for the introduction, hypothesis, summary,
Yeah.
conclusion. Do we have to each come up with that on our own, or will there be like an example?
no, what do you think?
I don't know.
Huh?
I don't know. That's,
Hey folks, George asked the following question. And the question is, when, hold on, everybody, please, pay attention. You've all been so good all semester. Now I've lost you. I'm gonna have to report to Dr. Bevensey. Yeah, seriously. Intro, hypothesis, strategy. Again, these are examples that are already uploaded, right? And then you have the large section results. And then the final section is summary, conclusion, acknowledgement, and references. Okay? Now, folks, this is your practice. This is where you put everything together. So, in answer to your question, George, use the PowerPoint that I've uploaded with a lot of introductory stuff on it, or read one or two papers and so forth on Bainbridge-Hoover syndrome, okay? And just generate this intro, right? Make sure you don't use anything less than a 24-point font. Yeah. Strategy, what's the difference between strategy and methods? Strategy and methods. Can you help me? What's the difference between strategy and method?
The strategy, like the methods are like your specific methodology, and the strategy is what, the methods are like your specific like methodology, like amounts used.
There you go. Very generic, and one is very specific. Thank you. All right, what else? I need to let you go because yes, we've devolved into badness. Except some. Here we go. Okay, any other questions? I'm gonna release your grades to you. Please, remember, you're gonna be graded on these presentations. Mr. Lamb, everybody got an 11 on lab notebook number 1. We did not grade you very hard. If you did everything, you got 11. Now, lab notebook number 2, it's not gonna be the same. Yeah, you miss one, you're gonna be deducted and so forth. And you have 5 or 6 people that will be grading you, and then 5 or 6 people that will be evaluating you. And I would even, I'll invite Dr. Bennett to come to your presentation. Yeah.
You used our data that we had to make the like example graphs and stuff. It's the same as what we're supposed to make. So would we
You can do the graphs.
use those graphs? That's
Would you wanna work at what time? Would you be available to work?
Or you the G418?
Yeah, with the G418.
Oh, you gotta go through it. You can't use my calculation. Yeah, make sure that you come up with the same stuff. Yeah.
I can also do Sunday.
AI.
I can do Sunday after we.
Hey, listen, folks,
Monday.
AI is not very bright. Yeah, it's only as smart as the operator. Try to ask AI to give you structure of amino acids, and it's gonna fail. Yeah, it's not that bright, okay? So always double check your work. Yeah, and so even though I feed it in, make sure that whatever is spitting out is correct, okay? Use a calculator, kind of check, double check, and so forth. Believe it or not, it makes mistakes. I review papers all the time, and I see faculty members submitting papers that have errors in them, because I know they have been using AI for analytics. It helps, but if you don't guide it, if you don't train it, it's not gonna help you that much. Okay? All right. What else? We good? Okay. I've answered all the questions, right? Hopefully you will do all the work, and I will see you on Tuesday. The sign-up sheet is gonna be uploaded today. We'll take a first come, first served basis. Yeah, you have to present, there's no excuse for missing presentation.
Do we not have class on Thursday?
We have presentation on Tuesday and Thursday.
Yeah, but
This
this
Thursday.
week.
No, this is your time to, please use it. Yeah, use it to prepare. Yeah. I can, listen, I can tell if you're just shooting from the hip, and that's not good. No, hold on.
Wanna meet at Domes or?
Everybody, hey, hey. Okay, please, this is very important. Mutant G418, right? They have 5 concentration from 0 all the way up to 400. Is that correct? You did 0, 25, and 50. They have 100, 200, and 400, right? You're gonna use all of it, 0 to 400, you're gonna use all of it. You're gonna do it separately, okay?
So we're just gonna, like, we're presenting separately, but we have
You
the
some-
same information?
Yeah, you're gonna use the same information, but you're gonna write it, you and your partner will write it separately, Yeah, you don't clone, I know if you just copy each other. Yeah, your conclusion is gonna be different, it's not gonna be the same. Yeah, Mr. Hamel?
But the figures are the same data.
Pardon me?
But the figures are all the same data.
The figures are,
Good.
Same data.
yeah, same data, yeah. But remember,
Don't
your
copy.
conclusion is not gonna be the same as,
Yes,
but
but
their conclusion.
can we share figures or not even